So let's basically talk about what happens when you have animal cells being placed in solutions of different tonicities. Remember, we have hypotonic, isotonic, and hypertonic solutions. So what happens when you place animal cells there? We start with a question. You have a student who's conducting an experiment on red blood cells. So the student places these red blood cells in 3 different solutions. 1, distilled water. 2, normal saline. 3, you have a concentrated salt solution. So can you classify these solutions according to whether they are hypotonic, isotonic, or hypertonic in relation to what you would expect to find in the red blood cells? Can you do that? So, tonicity refers to the concentration of the solids in the cell in relation to what happens in the external environment or the surrounding solution in this case. And we can have the 3 forms: isotonic, hypotonic, and hypertonic. And we are basically saying that if it is isotonic, the solid concentration is the same. So you don't have net movement of particles. In hypotonic, it means that the solid concentration inside the cell is high, and therefore water will move into the cell by osmosis. That is hypotonic. And then with hypertonic, the solid concentration in the solution is higher than what is in the cell. And therefore the cell will lose water to this hypertonic solution. So what could you expect? Part 2 is that if it is isotonic, basically you don't have any change in the shape of the red blood cell if it is isotonic. If it is hypertonic, that means that the cell will lose water and it will shrink basically. And when it is in hypotonic, it will take in water and actually burst. That's what you will see. Why? Because the red blood cell does not have a strong structure, does not have a cell wall. It has the cell membrane, and the cell membrane is not as rigid as the cell wall, and therefore it will burst. Okay, so can you compare the response of a plant and animal cells in response to hypotonic solution? We have just explained that plant cells will not burst, and then animal cells will burst. The red blood cells will burst in hypotonic solution. Can you suggest one reason why blood transfusions are done with isotonic saline instead of distilled water? Still, it's about the movement of what? Of water. You don't want to have water moving out of the cells into the bloodstream in this case, or even tissue fluid. You don't want that to happen. You want cells to be having the water that they have. So you don't want to create any disturbance in water movement. So that's why you need to have isotonic solution instead of distilled water. So like the question that we asked earlier on, so you would expect the concentrated salt solution is the hypertonic solution, and then the distilled water is water that does not have any impurities, so you don't expect any solid. So basically, this is pure water and has a water potential of 0. And then normal saline. So normal saline is basically made in that it depicts this, is actually osmo. It is isotonic. It should be having the same kind of solid concentration like the body fluids. That is normal saline. Should have similar concentrations of ions like our usual body fluids. Okay, that is blood and then some tissue fluid, because the tissue fluid actually comes from blood. Okay, so we have already explained isotonic solution, the red blood cell will remain normal because the water molecules are moving in and out at an equal rate. So you don't have net change in the cell's size or shape. And then in isotonic solution, it will take up a lot of water, swell and burst. Okay, and then with hypertonic, you will have it shrinking instead. Okay, are we clear? We can also demonstrate osmosis in living tissues, but we can also demonstrate osmosis using a membrane that is artificial, synthetic membrane. Cellophane is what we can use, and in this, it depends on the concentration of the solution that we can have inside the solvent membrane that is connected to the crystal funnel. And then we see the net movement of water and see what happens in the crystal funnel in relation to what happens in the volume of the solution that we have in the beaker. So the whole thing is that change the concentration of what is inside, and then either water will move in or will move out, depending on the solid concentration. That should be the logic. Okay, similar concept.